The next important cell type under the category of antigen-presenting cells are macrophages. Just like dendritic cells, macrophages are also an important part of the immune system, which plays a critical role in body's defense against infections, removing cellular debris, and contributing to tissue repair and regeneration. These versatile immune cells are found in various tissues throughout the body, where they act as vigilant sentinels, patrolling their territories and responding to signs of danger or tissue damage. If we look at their morphology, we find that macrophages are large phagocytic cells with an amoeboid shape. Also, they have an impressive ability to change their morphology to adapt to different microenvironments, including extending and retracting pseudopodia. This shape enables them to actively move and engulf pathogens and cellular debris. These macrophages also have extensions like dendritic cells, but their extensions are generally less numerous and not as long. Macrophages use pseudopodia that are the projections of the cytoplasm for motility and phagocytosis. Also, these macrophages are generally larger than dendritic cells, and their size can vary depending on their activation state and the tissue they reside in. Depending on their location and function, macrophages can be found as resident cells in specific tissues, thus called tissue-resident macrophages, such as Kupffer cells reside in the liver, alveolar macrophages in the lungs, and microglia in the central nervous system. These tissue-resident macrophages play critical roles in maintaining tissue homeostasis and immune surveillance, or they can be recruited from circulating monocytes during inflammation, where the mature monocytes are called macrophages. Monocytes travel through the bloodstream and can infiltrate various tissues. Upon reaching the tissues, monocytes undergo differentiation, transforming into mature macrophages with phagocytic and antigen-presenting capabilities. Then within the tissues, these macrophages perform plenty of functions. Where first one is phagocytosis, just like dendritic cells. So, one of the primary functions of macrophages is phagocytosis. And we know that it is the process of engulfing and internalizing foreign particles, such as bacteria, viruses, and cellular debris. And this process initiates same way, as we described for dendritic cells. As macrophages also possess toll-like receptors and scavenger receptors. And these receptors are able to recognize pathogen-associated molecular patterns, PAMPs, and damaged cell-derived molecules, DAMPs. Once the target is recognized, then macrophages surround and engulf them into the vesicles, called phagosomes. And then within another vesicle, that is phagolysosome, that is formed by the fusion of phagosome and lysosome, the engulfed material is degraded and destroyed by the action of lysosomal enzymes. So, to get the peptide of smaller fragments, just similar to what we have seen in dendritic cells. Now, this capturing of antigen is done by a similar manner as that in dendritic cells. But the antigen capturing by dendritic cells and macrophages exhibits significant differences as well. As dendritic cells are exceptionally efficient at capturing and internalizing a wide range of antigens, including pathogens, dying cells, and soluble antigens. But the macrophages are not as efficient as dendritic cells, and they primarily focus on engulfing pathogens and cellular debris present in the tissues. Next comes processing and presentation. Now, after phagocytosis, macrophages process and present fragments of antigens on their cell surface using major histocompatibility complex, or MHC, molecules, specifically MHC class II. The processed fragments of antigen gets loaded over the MHC molecule inside the cell. And then this antigen MHC complex goes to stand over the surface of macrophages as a tag, to be detected by other immune cells. This antigen presentation enables macrophages to communicate with T cells, activating the adaptive immune response and shaping the subsequent immune defense. Although both dendritic cells and macrophages are antigen-presenting cells, and once they capture antigens, they process them and then present them via MHC class II molecules at their surfaces. But the efficiency of antigen presentation of macrophages do not match with that of dendritic cells, as they are highly evolved for this purpose. And they mainly present antigen to helper T cells or B cells. But macrophages predominantly interact with pre-activated T cells, especially during secondary immune responses. They present antigens to effector T cells and memory T cells, contributing to the amplification and regulation of immune responses. Now, after phagocytosis and antigen presentation, the next important role of macrophages are in cytokine production. Other than APCs, macrophages are also potent secretory cells that release a wide array of cytokines and chemokines upon activation. And we know that cytokines and chemokines are the signaling molecules which acts as messengers, coordinating various aspects of the immune response. For instance, pro-inflammatory cytokines such as tumor necrosis factor alpha or TNF-alpha and interleukin-1 beta are released during infection or inflammation and help recruit other immune cells to the site of infection. Additionally, macrophages secrete anti-inflammatory cytokines such as interleukin-10 to regulate and resolve the immune response. Other than this, another important function of macrophages is in tissue repairing and regeneration. Macrophages contribute to tissue repair and regeneration by clearing cellular debris and promoting wound healing. During tissue injury or inflammation, macrophages shift their phenotype to become reparative macrophages or M2 macrophages. These M2 macrophages then produce growth factors, cytokines, and extracellular matrix components that aid in tissue remodeling and wound healing. Macrophages also help resolve inflammation, preventing excessive tissue damage and promoting the restoration of tissue homeostasis. Next, these macrophages play a vital role in immune regulation and tolerance, where immune regulation refers to the process by which the immune system is controlled and coordinated to ensure appropriate and optimum responses to various challenges. Whereas the term immune tolerance refers to the state in which the immune system does not mount an immune response against the body's own antigens or self-antigens. It means this mechanism ensures that the immune system recognizes self from non-self and avoids attacking its own tissues and cells. And macrophages play a critical role in immune regulation and tolerance by maintaining a delicate balance between immune responses and preventing excessive reactions against self-antigens. In the context of immune regulation, macrophages act as central coordinators. where they communicate with other immune cells and secrete cytokines to modulate immune activities, such as they can promote anti-inflammatory responses by adopting an M2 phenotype, which involves producing immunosuppressive cytokines like interleukin-10 and TGF-beta. This helps dampen inflammation and limit tissue damage during immune responses. Additionally, macrophages contribute to immune tolerance by promoting the development of regulatory T cells, which suppress excessive immune reactions against self-antigens. Also, macrophages in various tissues play a role in maintaining immune tolerance by engulfing apoptotic cells and presenting self-antigens to T cells in a non-inflammatory manner, promoting immune tolerance towards these self-components. So overall, macrophages are crucial mediators of immune regulation, ensuring an appropriate response to pathogens while avoiding harmful reactions against the body's own tissues. And lastly, their role comes in clearance of cellular debris. When cells undergo programmed cell death, that is apoptosis, or die due to injury or infection, they release their intracellular contents, including potentially harmful molecules. If not promptly cleared, this cellular debris can trigger inflammatory responses and tissue damage. So, macrophages that are equipped with specialized receptors recognize specific "eat me" signals exposed on the surface of dying or dead cells that are usually called DAMPs. So, once they recognize these DAMPs, again the process of phagocytosis begins, and thus they internalize these apoptotic or dead cells, effectively removing them from the tissue. Then inside the macrophages, lysosomal enzymes degrade the engulfed cellular components, neutralizing any potential harmful materials. In this way, it helps clear the cellular debris. And thus, at the end, no matter whatever the antigen is, captured, processed, and then presented in bound form with MHC class II molecules at their surface. And when this tag encounters the CD4 plus T helper cells, they becomes activated and thus coordinate immune response against the captured pathogen. So, this was all about macrophages in context of antigen-presenting cells, where we learned how macrophages perform various immune functions, where the most popular one is antigen capturing, and then its presentation to the subsets of T cells to generate appropriate immune response. Ever felt overwhelmed by the traditional way of learning medicine? Enter the future of education with digital learning. Whether you're refining your knowledge of biochemistry or immersing in cell biology, it's all there. Understanding complex medical subjects has never been easier or more efficient with engaging, interactive content. Welcome to the new age of medical education. Learn anywhere, anytime, at your pace. Powering your medical journey. sqadia.com